Dear students, welcome back to the channel Kingdom Zoology. Let's continue forward with our studies of biotechnology. What we have covered in various lectures in biotechnology till now that you can see on the channel among the under the playlist class 12th biotechnology and otherwise also is the introduction to the biotechnology. That is the first thing that we have done till here in this chapter. What exactly is biotechnology? What is genetic engineering? Why do we actually need to isolate some desired genes? How do you actually isolate or obtain them? So all that came under introduction chapter. And of course, in the previous lecture, I explained to you what exactly are various types of restriction endonucleases, what exactly is their role in trying to identify or purify or isolate a desired gene. So for clarity on all these topics, you can always refer to the previous lectures shared on the channel on biotechnology. So today we will be going forward with our type 2 restriction endonucleases, their examples, and the technique called gel electrophoresis. That's another very important technique for NEET exam because in NEET 2025 also we had two questions from gel electrophoresis itself only. So let's continue then. So about type 2 restriction endonucleases, these were the types of restriction endonucleases that identify a particular palindrome, which means इनका जो mode of action है वो क्या है? If they have to digest a genome, they will first need to attach to that genome. On the genome, they need to digest. So where do they attach? On the particular palindromic recognition sites. The palindromes can also be called their recognition sites. So they will have to first attach. And then the most important point about type 2 enzyme that differentiates them from the type 1 and the type 3 enzymes was that they cut the phosphodiester bonds on the genome within this palindromic recognition site. So that is one thing about them, that they cut the genome within this palindromic recognition site, and that too at a fixed location or point. Which is why if I have obtained the genome of a cell, this is the cell who's the natural producer of our desired protein product, the protein product that I want to produce at a mass commercial scale for the benefit of the humans. So I took out the genome of this cell. Now the genome needs to be digested by type 2 restriction endonucleases so that it would be broken into small pieces. And if let's say one of these pieces is the one that is going to code for your product, we discussed in the previous lecture that I might require more copies of this thing. So if I need more copies of this thing, I'll take the same cell, take out the same genome, put the same enzyme, and I would expect that I should get the same fragments. That is only possible if I'm using type 2 restriction endonucleases because if I use type 1 or type 3 restriction endonucleases, they also identify specific palindromic recognition sites on the genome that they need to digest. But the problem with them is they don't cut at specific sites. The cutting sites are far away for type 1 enzymes, still away from the recognition site, and never fixed in case of type 3 enzyme. The advantage with type 2 enzymes was they will identify a particular recognition site, a palindrome. They will attach over there and they would cut within that palindrome, always at a fixed location, so that the fragments that you get by digesting a genome with a type 2 enzyme in one go, in first attempt, the exact same fragments will be produced during repetition of the experiment, during trying to cut the same genome by the same enzyme next time also. So you'll be always getting the same fragments. So you can be sure of getting the same results or the same gene fragments from a genome only if you are using type 2 enzyme. That is why we chose type 2 enzymes for our biotechnological procedure. Now, what we'll be doing today is that within type 2 restriction endonucleases, there are two different choices. Okay. The type 2 restriction endonucleases are themselves of two types. These two types are blunt, also called flush end producing enzymes, and the second is sticky or cohesive end producing enzymes. There's one thing that is true about all the type 2 enzymes. They will attach to a particular palindrome by identifying it. They digest the phosphodiester bond in the genome always at a fixed location. But then the type of cut they introduce on the genome, that would be actually different. And on that basis, the type 2 enzymes themselves are of two types: blunt end producing enzyme, sticky end producing enzyme. Let's see what this means. Let's imagine I have a genome to be digested, which can be represented like this here. Here's the 5' end. That's basically your genome that you need to digest. A long sequence of nucleotides because it's the entire genome, right? So this is what I need to digest. And since it's the DNA, the 5' and the 3' ends, of course, are what? Anti-parallel to each other. So here's the representation of the genome to be digested. Now in this genome, let's say there's a sequence that is A, G, C, T. Here again, if you look at the complementary bases, T का complementary कौन है? A. C का complementary है G. G का complementary C. A का complementary T. So can you see that this is actually what? A palindromic sequence. And it's not compulsory that the palindrome will repeat only once in a genome. It can repeat any number of times. So let's say there is one more place where the same palindrome is repeating almost near the end here. So the same palindrome is actually repeating in this genome two times. Now I put a type 2 restriction endonuclease over here. This type 2 restriction endonuclease is going to identify this palindrome and attach to it. Then the enzyme is going to cut the phosphodiester bonds between G and C. These are the points where the phosphodiester bonds will be digested. And then the enzyme will shift and will find another recognition site for itself and does the same. Digests this and this phosphodiester bonds. So when these phosphodiester bonds are digested, they will be cut here, cut here. This one, this one. These phosphodiester bonds are cut. So can you see that your genome has now been broken into how many fragments? 1, 2, 3. The type 2 restriction endonuclease that does this activity, the one that identifies this AGCT palindrome, the name of that enzyme is ALU1 enzyme. And the gene fragments produced by this enzyme are going to be, let's see what. There'll be 3 fragments produced by it. So if we look at the genome, which was digested by this enzyme, it'll break into three fragments. So I would just draw all of them first here. And we can actually then understand where exactly the cuts are introduced. So here, this base is A, this is G, that's a C and that's a T. Once again here TC, GA. So can you see that here the genome has been cut and has been converted into what? Two different pieces. Similarly, if we go ahead, here's A, G, here's C and T. Here's A and G, here's C and T. So once again, when I introduce the cut over here, AG, CT goes into what? Next fragment. So there will be 1, 2, and 3 fragments created. But if you look at all these three fragments, all the three fragments actually have been produced by cutting the DNA strands opposite to each other. The cut has been introduced at this place and exactly opposite over here. At this place and exactly opposite over here. So as a result, the three fragments that we have got, 1, 2, and 3, these three fragments, none of them have got any single stranded portion. They are complete full double stranded DNA. So this type of gene fragments, which do not have any single stranded portions, complete double strands, these are called the blunt ended fragments. These are the blunt ended fragments, and this type of type 2 restriction endonucleases, which are producing these kind of fragments, are called blunt or the flush end producing enzymes. So that's your types of restriction endonucleases. One category, the blunt or flush end producing enzymes. Now let's move on to the next type, the sticky or the cohesive end producing type 2 restriction endonucleases. Once again, let's first mention the genome to be digested. Take an example of that. Genome to be digested. So let me draw that again here. Just a representation once again. Yeah. So in the genome to be digested here, we would be having, let's say, another representation. A double-stranded DNA like this. Now, in this genome, let us draw a different type of palindrome, maybe. Let me begin from here. Let's say GAA TDC. Yeah. And the opposite strand, GAA TTC. Then maybe same thing after, let's say, a distance. Once again, AATTC. On the opposite side, You'll have the same sequence G, A, T, T, C. So this time I'll add a type 2 restriction endonuclease on this genome whose name is EcoRI enzyme. Now this recognition site or this palindromic site is the recognition site of which enzyme? EcoRI. So when I add EcoRI enzyme to cut this genome, EcoRI is also going to introduce cuts, but where? Here at this point, and same over here at this point between the G and A. See, it is attaching to a particular recognition site and then cutting within the recognition site. So what type of enzyme is it? Type 2 enzyme. But then compared to this one, which was introducing cuts exactly opposite to each other. This enzyme is not cutting the phosphodiester bonds exactly opposite to each other. It's cutting one strand over here and the other strand over here. Then after doing this cut, the enzyme is going to shift forward, reach over here, and after reaching over here, it is going to do the same thing, introduce cut between G and E, here, and of course, here. These are the places or the sites where the phosphodiester bonds will be digested. So here, the gene fragments produced by the enzyme are going to be what? Let us see what will be the gene fragments produced by the enzyme in this case. Let me draw a line over here between the two examples. These are your blunt end producing enzymes that have produced blunt ended fragments. And on the other side, now we'll see how the sticky end producing enzymes produce different types of ends. Here, let's see. The first segment that I'll get here is beginning from the 5' end, ending at G only. Why do you think the strand is going to end at G? Because the bond between G and A has been cut. But if I look at the complementary strand on the lower side, which starts with the 3' end, here is C attached to G. These two bases are also hydrogen bonded to each other. But if you look at this strand, will it end at C? No, because C is attached to T, that's attached to one more T, that's attached to A, that's attached to A. Here, the strand is cut. So the lower strand is going to basically stretch a bit more. There'll be a T here, T here, A here, A here. That'll be one fragment that has been produced. Now look at the second fragment. The second fragment, so if we look at this picture, here is your one fragment. The second fragment will be this one. This will be your second fragment. And then this will be your third fragment over here. So how many fragments have I got again? One, two, three. But can you see that each fragment has got some overhangs on it? These overhangs are called the sticky or the cohesive ends. Let's just first draw them. This was your first strand. The second one begins with A, A, T, T, C, and then in the end again, this is going to be the end of that second fragment on the G, because the bond between G and A has again been cut. So that's G. If I talk about the complementary strand on the lower side, that would be beginning from here. There will be G over here, and then there is C, T, T, A, A. Can you see what I've done here? AATTC, AATTC, ending at G. But the lower strand starts with G because these are parts of the previous fragment. So starting with the G here, going up to A, going up to A, that would be your middle fragment. And what about the third and last fragment? Let's see again. That's the 3' end, and that's the 5' end here. So if I start from here, what is the strand end at? G. But what about the upper strand? Where does this end at? A. There's going to be G over here attached to C. So this is your third strand, fragment. And if I complete this portion here, this would be C followed by T, T, A, A. That's it. Here are your three fragments. So if I look at fragment number one, in this case, this is your first gene fragment that has been created with a slight single-stranded overhang. That's your second fragment which has been created by this enzyme with single-stranded overhangs on both the sides. And that's your third fragment with a single-stranded overhang over here. So in this case, if I just join these things together, join this over here, join this over here, what do I get again? Same thing. So what happened in this case is the enzyme broke down your genome over here also into how many fragments? Three. Because of introduction of cuts at two places, 1, 2, 3 fragments are produced. Here also the cuts are introduced at these locations. But if I cut this strand here and here, and but the lower strand over here and over here, you're going to get one fragment, then the second fragment, and then the third fragment. So fragment number one, fragment number two, fragment number three. But this has got an overhang. This has got an overhang on both the sides. This also has got an overhang. All of them have got sticky ended overhangs. Why are they called sticky ended overhangs? Because these are free nucleotides. They want to attach to something. And what will they attach to? Any other DNA which has got complementary base pair sequences on it. So since these are single stranded overhangs, what do we also call them? Sticky ends. So here, these are your sticky ended fragments. Sticky ended fragments with single stranded overhangs on all the fragments. These single stranded overhangs, what are these called? The sticky ends. So that, guys, is the second type of restriction endonucleases type 2, that is sticky end producing enzyme. So once again, just a quick recall. Once you isolate the genome from the cell that codes for your desired protein product, you have to break down that genome into small gene fragments, and for that you require restriction endonucleases. Restriction endonucleases have got three choices: type 1, type 2, and type 3, out of which these are the ones that we choose because their special properties, identifying a palindrome and cutting within that palindrome, always producing exactly same fragments every time a genome is cut by using these enzymes. But within type 2, I've got two options here: blunt end producing and sticky end producing enzymes. So you have, if I have isolated a genome and to cut it, I have put type 2 restriction endonucleases. I can see that there are two options in the type 2 restriction endonucleases: blunt and sticky end producing enzymes. Even though both of them can be used, if you have to choose one of them, these or these, what do you think will be the better option? So you can actually tell that to me in the comments when you are done watching this video. Let's have some more discussion further about these. The sticky end producing enzymes. Sticky end producing enzymes are preferred in biotechnology. In the process of digestion of genome. The sticky end producing enzymes are preferred in biotechnology in the process of digestion of genome. Why? Just a simple idea. This is your genome that has been digested into how many fragments? Three. Let's imagine this is the fragment that is going to code for your, that has the capability of coding for your desired product. So if this is my fragment that can code for the desired product, I'll have to put it into what? Someone like maybe a bacteria. So here is the bacteria chromosome, and I have to basically attach my desired gene also over here. So let's say if the bacterial chromosome also has got blunt ends and your desired gene also has got blunt ends. So we'll attaching your desired gene to the bacteria's genome. Which bacteria is it? The one that will be producing your desired product at the mass commercial scale, because you've got the desired gene now. All you need to do is add it on maybe the genome of a cell like a bacteria, which will be producing your desired product for, I mean, human use at commercial massive scale. But the problem over here you can understand is if your gene is blunt ended and the genome of this organism where you want product formation to occur is also blunt ended, attachment is not going to be easy. However, if I have this bacteria and I can actually cut the genome of the bacteria in this kind of a way that it would be looking something like this. That is the genome of the bacteria, which has been cut in this way. Here's the bacterial genome. Now, what does the bacterial genome contain over here? Overhang. What does it contain over here? Overhang. Now, if I want to attach this fragment over here, this overhang can attach over here. This overhang can attach over here, and the intermediate part would be over here. So can you see that the attachment of your gene fragment with the genome of some other cell will be easier if it has sticky ends. Let me show you in an actual refined diagram. This is just a vague idea that I gave you. The sticky and producing enzymes are preferred in biotechnology in the process of digestion of genome because the sticky ends created by them on the gene fragments. The sticky ends created by them on the gene fragments will help in easier ligation or attachment of the desired genes. This is the desired gene, and I need to attach it to some other genome. The sticky ends created by them on the gene fragments will help in easier ligation or attachment of the desired gene fragments with some other DNA of some other cell who will be used as the factory, kind of, you can say, for mass scale production of the desired product. If this is your fragment that codes for the desired product, or this is your fragment that codes for the desired product, attaching which one of them to some other cell's genome. Which cell's genome? The one that will be actually using as a factory for mass scale production of your desired product. So attachment of which of them will be easier to the genome of the other cell, which will be used as a factory to produce desired product at a mass scale. Obviously these ones. Let me show you how. If the desired gene is blunt-ended, here's your desired gene, which is going to code for the desired protein product, the protein product that you want at a mass commercial scale. If the desired gene is blunt-ended, it can be added on the genome of another cell, the one that will be used as a kind of factory to produce large amount of products from this desired gene. It can be added on the genome of another cell with a lot of difficulty. It's not easy to actually attach this kind of fragment. Here's the cell in which the desired gene needs to be expressed. And here is the genome of this cell. And I want to attach this blunt-ended fragment over here. So it's okay, I can just put it over here. The blunt-ended fragment has been put over here in the genome of the cell where you want the gene to be expressed and the product to be formed. So the problem over here is, what is exactly linking these two? Nothing. So what are you supposed to do? Add a large amount of DNA ligase enzyme. And once you add a large amount of DNA ligase enzyme, then only these phosphodiester bonds will be formed. And then only you'll be able to attach your blunt-ended fragment to the genome of the cell so that this can be expressed and products can be formed at a mass or a commercial scale. So this is the situation that if you have blunt-ended desired gene and you have blunt-ended genome of the cell in which you want to add this gene for commercial mass scale production of the product. The problem with the blunt-ended fragments is they are not easy to ligate or attach to some other genome of the cell that will be used as a factory for production of your desired product at a mass scale. So let's see the other option. If the desired gene is sticky-ended, if it has sticky ends, if the desired gene is sticky-ended, it can be represented like this. That's your desired gene with sticky ends like this. Single-stranded overhangs are called what? Sticky ends. It can be easily added on the genome of another cell where the expression of this gene is required. How? Let me tell you. This is now your cell used for expression of the desired gene. And what do we have here? The genome, sorry, the gene fragment that will be coding for the desired product. So now what are we supposed to do? Now what we are supposed to do is cut open the genome of this cell in this way. That is how the genome is cut now, with single-stranded overhangs on it. Now you see, if this single-stranded overhang is complementary to this one, and this is complementary to this one, तो हो गया काम, easily attach हो जाएगा. But यहाँ पे did you notice what I just said? I said that if ये तो double-stranded portion हो गया बीच का, that is this portion here, ये हो गया यहाँ पे attach. But ये वाले overhang को इससे अटैच करना है और ये वाले ओवरहैंग को इससे अटैच करना है. तो जो ओवरहैंग्स के ऊपर बेसिस हैं वो सभी आपस में क्या होने चाहिए? कॉम्प्लीमेंट्री. लेट्स सी हियर. ऑन दिस साइड एंड ऑन दिस साइड. सेम बनाया ना जो यहाँ पे था, यही दोबारा से यहाँ पे बना दिया. बट लुक एट द सिचुएशन नाउ. Will there be hydrogen bond formation between the sticky ends of the genome of the cell? which will be used as the source of production of our desired product. तो उसका खुद का genome and हमारा desired gene. Do you think it's easier to attach in this way? Yes. Why? Because see, even though DNA ligase तो यहाँ पे भी required होगा. What is the DNA ligase required for? To form the phosphodiester bonds over here. तभी पूरा complete double stranded DNA बनेगा. तो ligase तो यहाँ पे भी required होगा. One. Look at the zoomed in picture. 1, 2, 3, 4. These are the 4 places where phosphodiester bond formation is still going to be needed. तो उसके लिए तो डीएनए लाइगेज लगेगा, बट द पॉइंट इज जितना लाइगेज का क्वांटिटी आपको इधर चाहिए था उतना नहीं चाहिए. लाइगेज नहीं भी होगा तो भी, इवन दो इट्स नॉट अ कंप्लीट डबल स्ट्रैंडेड डीएनए, बट स्टिल ये है कि भाई इसमें भी स्टिक तो रहेंगे एक दूसरे से सिंगल स्ट्रैंडेड ओवरहैंग्स होने की वजह से. ठीक है, सो रीड दिस अगेन नाउ. There are two types of type 2 restriction endonucleases, blunt end producing and sticky end producing, out of which sticky end producing enzymes are preferred because अगर आपने enzyme use किया blunt end producing तो आपके पास ये desired gene है, इसको किसी और cell के genome से attach करना होता है बहुत difficult. बट अगर आपने एंजाइम यूज किया था डिजायर्ड जीन को ऑब्टेन करने के लिए स्टिकी एंड प्रोड्यूसिंग एंजाइम, तो आपका डिजायर्ड जीन है ये विद द स्टिकी एंड्स ऑन इट, और इसको अटैच करवाना किसी और सेल के जीनोम पे जहां पे आपको इस जीन की एक्सप्रेशन करवानी है. तो इस जीनोम को सॉरी इस जीन को अटैच करना किसी और सेल के जीनोम पे इज कंपैरेटिवली easier. That is why out of the two options that you have in the type 2 restriction endonuclease category, which ones do we actually choose? The sticky end producing enzymes, not the blunt end producing enzymes. But then remember this important thing that I said in between, that you can easily attach a sticky ended gene fragment to another cell's genome, but the condition is the sticky ends present on your desired gene, I mean the gene that will be coding for the desired protein product, and the sticky ends present on your cell's chromosome, दे मस्ट बी कॉम्प्लीमेंट्री टू ईच अदर. अदरवाइज तो कोई मतलब ही नहीं है ना स्टिकी एंड का. मान लो इसके स्टिकी एंड कुछ और है और इसके स्टिकी एंड कुछ और है, आपस में कॉम्प्लीमेंट्री नहीं है, फिर थोड़ी ये अटैचमेंट होगा यहाँ पे. दिस अटैचमेंट हैपेंस ओनली इफ द sticky ends of your desired gene fragment are complementary to the sticky ends present on the cell's genome. Okay, so that is the limitation. Otherwise, sticky end producing enzymes are always better. And if you talk about the examples of enzymes producing these ends, examples of enzymes producing blunt ended fragments, पहले लिख लेते हैं इस तरफ. Examples of enzymes producing blunt ended fragments. Examples of enzymes producing blunt ended fragments. एक तो ऊपर एंजाइम लिखा ही है ALU1, और क्या एग्जाम्पल है इसमें SMA1. ALU1 कौन से पैलिंड्रोम को आइडेंटिफाई करता है? AGCT. AGCT. तो ये पैलिंड्रोम आइडेंटिफाई कर लिया AGCT. इसमें G और C के बीच में क्या लगा दिए? कट. तो क्या बन गए? दो फ्रैगमेंट्स, दोनों ब्लंट एंडेड हैं. One more example is SMA1. Let's see what is SMA1. What is this cut? It's triple G, triple C. So here, this is your palindromic recognition site of SMA1 enzyme. तो इसको आके अटैच कर लिया एंजाइम ने और कट्स इंट्रोड्यूस करने हैं कहाँ पे, सेम दोनों स्ट्रैंड्स को आमने सामने, इधर और इधर, सो दैट आई वुड बी गेटिंग वन फ्रेगमेंट, सेकेंड फ्रेगमेंट। वंस अगेन बोथ द फ्रेगमेंट्स आर व्हाट, ब्लंट एंडेड। तो ये साथ में हमें क्या पता चल गया इस डिस्कशन से, द टाइप ऑफ recognition sites of some common blunt-ended producing enzymes. And if you look at the examples of sticky end producing, examples of enzymes producing sticky ended fragments. Examples of enzymes producing sticky ended fragments. One example is EcoRI. The example that I've shown you above with the recognition site GAATTC. ये एक्चुअली में किसका साइट है, EcoRI का। EcoRI whose recognition site is GAATTC. And the cutting site is the bonds within G and A. So that what do you get? What do you get? Sticky-ended fragments. So that is one example of sticky-ended producing enzymes. The other example can be BamHI. BamHI is the enzyme whose recognition site is GGATCC. Here, G, G, A, T, C, C. So can you see this is a palindrome? Palindrome is a sequence that reads the same on both the strands. GGATCC, GGATCC. This is also a palindrome. And where exactly are the cuts introduced in this case? Between the two G's, so that there will be one fragment and there will be the second fragment, with these being the single stranded overhangs. So दोनों ends क्या बने, sticky ends. So EcoRI, BamHI are sticky ended producing enzymes. ALU and SMA1 are blunt end producing enzymes. Okay, and out of these, as we just discussed, the ones that we prefer are going to be sticky ended producing enzyme, because attaching the sticky ends to some other genome, which obviously anyways has to be done because we want the desired gene to code for products. How does it code for product? By going into some cell. Which cell? The host cell in whose genome we have to attach it. So attaching this fragment versus attaching this fragment with an overhang, obviously this will be much more easier than attaching a block without any overhangs. Okay, so now after this, we also have a small discussion that needs to be done about the rules of nomenclature of the restriction endonucleases. So for now, what we have done is how the restriction endonucleases type 2 work. They can either produce blunt ends or they can either produce sticky ends. Which one do we prefer? So we will wind up here today. In the next series or the next lecture, I would take this discussion forward with respect to what exactly are the rules of nomenclature of the enzymes. And once we will understand that, then we'll move on to the next part. That is, once you have broken down your genome into various fragments, we need to separate the fragments on the basis of their sizes and then identify the correct fragment that will be coding for your desired product. So for all this, stay tuned. I'll keep sharing more videos after this. The today's discussion comes to an end over here. Thank you for joining. Keep liking, subscribing the channel. Keep sharing it with your friends. Sharing the knowledge is not going to basically increase your competition. It is actually going to help you in the longer run, because the more like-minded people you compete with, more better prepared you will be for the NEET exam. Okay, thank you everyone. See you in the next lecture.